Hi, my name is Miriam Sun. I am a graduate student in the O'Brien Lab at Stanford University. And thank you for the opportunity to speak at today's Physics of Life Symposium on how stem cell clustering can influence fate decisions during injury. So to first set the stage, adult stem cell-based self-renewing organs must respond to unpredictable insults throughout an animal's lifetime. During homeostasis and regular cell turnover, when a mature cell dies, stem cell division must be coordinated with appropriately paced differentiation in order to produce a new mature cell replacement. This invokes a sort of global cell balancing act where these local processes are all tied to each other so that the organ can maintain the right overall proportions of these different cell populations. But what happens when the organ is injured? During injury, more mature cells die all at once than would normally occur during healthy conditions. And we know from the literature that organ stem cells divide more during injury, which makes sense as a response to the acute need for new cells. But what the tissue really needs is not a bunch of new stem cells, but new differentiated functional mature cells to replace the ones that were lost. What I want to focus on today is on this process of differentiation from stem to terminally fated progeny and eventually mature cell, and on what happens to progenitor cell interactions in order to drive terminal fate decisions without depleting the stem cell population, but still matching the injured organ's new needs. Now, in the O'Brien lab, the stem cell-based adult organ that we use for our studies is the fruit fly gut. It is a single-layer epithelium, has a simple fate lineage from stem cell to terminal progeny or enteroblasts, and finally mature cells or enterocytes. It is easily genetically tractable, amongst other great advantages of the Drosophila model organism. Now, fate decisions in the fly gut, like in many other systems, is governed by delta notch signaling. This cell-cell contact-dependent signaling follows a process termed lateral inhibition, standard equations of which are shown here. So two initial progenitor cells, both expressing delta ligands and notch receptors, signal to each other when in contact. Over time, small initial differences in signaling compound and break symmetry, with the resulting notch-on, delta-off cell being the terminally fated enteroblast, while the cell that remains notch-off stays a stem cell. In this way, delta notch signaling promotes opposing cell fates and drives fate decisions in the fly gut. Here's an image of what the fly gut epithelium really looks like, with these smaller progenitor cells interspersed amongst the larger mature enterocytes whose cell outlines are marked in gray. In the green channel is a reporter of notch activity, or NRE, driving GFP that we use to identify the notch-on enteroblasts. So we first started by looking at both delta expression and notch activity in healthy versus injured guts to see how this fate-determining signaling circuit might have changed during gut injury. Here is an image of healthy gut progenitors, this time without the other cell outlines. And you can see that the purple stem cells and the green enteroblasts are interspersed throughout the epithelium. And as expected of delta notch lateral inhibition, the stem cells express delta, shown here in blue, while the terminally fated notch-on enteroblasts have turned off delta. And you might also notice that these progenitor cells are often arranged in pairs, but rarely more than two are in contact with each other. Now, when we look at an injured gut, immediately you can notice some notable differences in the tissue arrangement. First of all, the green notch-on enteroblasts now also express a blue ring of delta expression, indicating that delta notch lateral inhibition has been disrupted and delta is no longer being turned off after the cell turns on notch. Secondly, and perhaps more obviously, in the injured gut, there are many multi-cell clusters of progenitor cells with 3, 4, even 5 or more stem cells and enteroblasts clumping together and staying in contact with each other. From this, we conclude that 1, injury disrupts delta notch signaling feedback, and 2, injury induces a clustering effect of the newly generated progenitor cells. To better understand the effect of these two changes on delta notch signaling and cell fate decisions, we turn to mathematical modeling. In collaboration with fellow Stanford graduate student Salman, we again based our model on the standard equations for lateral inhibition, where the active notch level of a cell is an increasing function of the delta level of its neighboring cells, and the delta level of a cell is a decreasing function of the active notch level in the same cell. And I want to focus your attention on two particular terms. The first one is this KD term, which describes the threshold of delta being inhibited by notch activation in the same cell. And we previously saw that in injured guts, the notch-activated enteroblasts were no longer turning off delta, which suggested that KD is increased. And if you're curious how increasing KD and delta expression during injury accelerates stem cell differentiation and gut organ repair, please check out our preprint out on bioRxiv and currently in revision. But today, I want to focus on a story in the early stages about this other term, Kn, which describes the threshold of notch being activated by delta on neighboring cells. Kn is thus inversely related to the contact area between two cells. It can be influenced by how many cells are in contact with each other and signaling to each other at the same time, or even affected by cell-cell movement and evolving cell interactions, all of which is where that second progenitor cell clustering phenotype really comes in. In collaboration with BioHub scientist Zubin from the Jacobo group, as well as with the help of visiting master's student Sebastian, we quantified the distribution of cluster sizes in healthy versus injured guts. And you can clearly see where in healthy guts, about 90% of progenitors occur in singlets or in pairs. In injury, however, there is a significant shift towards cluster sizes of 3 cells or more. And now looking at cluster sizes side by side, we see clearly that when more progenitor cells are in contact with one another, a larger percentage of that cluster is made up of notch-on enteroblasts. Further breaking down a cluster size group by its composition of stem cells versus notch-on enteroblasts, what becomes apparent is that in injured guts, more of the cells in any one cluster size are terminally fated notch-active enteroblasts. For example, looking at cluster sizes of 2, or pairs of cells, we see that in injured guts, there's about a 25% increase of enteroblast-enteroblast pairs and a 5% decrease of stem cell pairs. This trend holds for clusters of 3 cells, where triplets of stem cells or of 2 stem and 1 enteroblast are reduced, while triplets with 2 or all enteroblasts are increased in the injured condition. And this pattern generally holds as cluster sizes continue to increase. So now that we have this rich imaging data on progenitor cell fates in healthy versus injured organ states, can we construct a model that can first reproduce our in vivo phenotypes, and secondly, be used to ask how organ scale fate outcomes and repair emerges from these local fate decisions? Our goal is to eventually be able to factor in population level processes such as stem cell division rates, differentiation rates, and also the aggregation or breaking up of these progenitor clusters, since progenitors are capable of moving through the epithelium. And these parameters will be informed by high-resolution live imaging data that we are working to acquire more of, that can tell us approximate rates of cell exchange between clusters, as well as change in contact areas over time. And lastly, I want to show some preliminary simulations where we have these little simulated progenitor clusters spread out in fixed spots across the in silico gut epithelium. Here we have the cells instantaneously exchange between clusters. But so in the simulated healthy gut, we see that there are few clusters of more than 3 cells. But when we test parameters that are more aligned with an injured gut state, such as more cell death, higher KD, and faster differentiation, in line with our in vivo observations, in the injured gut, there is much more clustering of progenitors, and the larger clusters of progenitors do tend to be made up of more of these green notch-on cells, suggesting that the particular spatial arrangement of signaling progenitors that arises during injury is in fact instrumental to promoting terminal cell fate and timely organ repair. And I will end with my acknowledgements. Once again, thank you to everyone who has worked on this project. Thank you to our funding sources, and thank you all for listening.
All right, we have some time for questions. One here.
So what do you know, or what's your intuition about so the signaling pathways and cell-cell interaction modalities that will result in increased clustering or the breakup of clusters? Are those acting through independent signaling pathways that are independent of Notch and Delta, but then the behavior of the cells has an impact on Notch Delta signaling? Or is there communication between Notch Delta and things affecting cell behavior?
Right. So what we do know is that in injury there are some stress response signaling pathways that are also activated that drive things like increased cell proliferation that we think can initially cause more progenitors to be in close contact with each other at first, and that does influence delta notch signaling since it's a cell-cell contact dependent signaling factor. Interestingly though, in injury in live imaging movies, we also see that there's more cell movement. So we are kind of working on figuring out like how all these different factors fit into each other, like the relative effects of each. So we're looking at, knocking down different factors in different pathways or cell movement factors, and to really nail down the mechanisms. Yeah.
Hi, thank you. It's interesting talk. Maybe I missed this, but can you give a little bit of intuition for why the clustering that you observe, how that addresses the initial conundrum you posed of how you ensure that when the tissue regenerates, you kind of have that right balance of differentiated cells versus adaptable stem cells?
Right. So what we knew initially was that when the tissue is injured, you get a lot more stem cell division, so you have more stem cells. But the stem cells, if they're just kind of clumping together by themselves without differentiating, that doesn't really help the tissue very much, I guess. And based on delta notch cell-cell contact signaling, when there are more of those progenitors that can signal to each other in contact, that tends to drive more signaling, more notch being activated, and more terminal fates being driven. And today I mainly showed like 2-cell models of lateral inhibition, but we have some work that we've done expanding it to 3 cells or more, where looking at more progenitor cells interacting with each other at once, like a higher proportion of those progenitors in a one cluster will then activate notch and become differentiated. So that's why we think that the progenitors staying in contact with each other is pivotal to driving more terminal fates and thus repairing the organ.
I think we gotta wrap it up. Find Miriam afterwards. Thanks again, Miriam.